All right. My name is Peter Bross. I'm from the Center for Biologics. And when I started at FDA, I had a room right across from Raji in Cedar. And let's see if I can get this to work. I have nothing to disclose. I work for you. That's where I work. I'm going to talk about this is an outline. I'll talk about FDA regulation in general, and specifically about our office and Office of Cellular Tissue and Gene Therapy. And then I'll talk a little bit about the regulatory challenges of personalized therapy and combination therapies and certain regulatory initiatives, and then contact info if you have questions or complaints. This is a little bit about the structure of the FDA regulation of oncology products, and I will say that there may be some changes afoot with the President's Moonshot initiative. We'll see. We may all be regulating from the moon. And when I started out in Center for Drugs, as I said, I was across from Raji, and we regulated small molecules, and there were some exciting small molecules I got involved in. And just as things got interesting and the monoclonal antibodies and protein cytokines came over there, I moved over to Center for Biologics. I thought it would be peaceful and calm and everybody would be sleepy, but it turned out it was a pretty interesting place. We regulate cell therapies, gene therapies, oncolytic viruses, which we recently approved one, therapeutic vaccines as opposed to the preventive vaccines, which are regulated in a different office in CBER, and also certain devices. We regulate the Clinimax, and I actually did a review of an apheresis device. And last but not least, Center for Devices, which includes the Office of In Vitro Diagnostics, which regulates companion diagnostics, and then the Office of Device Evaluation, which regulates therapeutic devices. And I won't be talking about them, except for companion diagnostics are important in terms of personalized medicine. This is the office structure. I work in the oncology branch, and I'll say the PharmTox branch, where Alan Wensky works, is very busy. This is another review of our regulatory scope. I'll focus on cellular therapies, cancer vaccines, oncolytic viruses, and gene therapies. And as I mentioned, we also regulate certain devices. So we're sort of jack of all trades in a way. So I'm going to focus on personalized immunotherapies and divide them maybe artificially into three basic types. I'll talk about some that are based on the biomarker target, which is probably familiar to everyone, such as EGFR, KRAS, MUC1, or CD19. And these may require a companion diagnostic, and also mention that some of our products require HLA restriction, and this, just to confuse everybody, is regulated in the Office of Blood. Anyway, the second type, it would be a neoantigen-based manufacturing of actively personalized products to treat individual patients, which Dr. Singh talked about earlier, and I stole one of his slides, you'll see. And the third type is based on autologous manufacture. And a long time ago, people were mixing up tumor antigens. They would lyse tumors and then use these as cancer vaccines. Then came along the dendritic cell manufacturing, and then most recently, the so-called designer T cells, including the chimeric antigen receptor T cells that Dr. Porter talked about. So to begin with, biomarker targeted therapies, ones that we are have been involved in include targets against MUC1. There was a lipopeptide that was used in non-small cell lung cancer, unfortunately was not successful. MAGE-A3 was also a peptide vaccine for non-small cell lung cancer and melanoma. Apparently, unfortunately, that didn't work out so well. And now we have an EGFR protein conjugate that was actually being imported from Cuba, and we'll see how that goes. So hopefully these are close to no cigar. Maybe they'll get a cigar on the on the new one, but studies are ongoing. It's very interesting. We have to figure out if it's legal to import those things from Cuba. I think it is. Now you'll recognize Dr. Singh's slides here, which I borrowed shamelessly, just to make the point of the different types of active personalized, the different types of personalized medicine. The first would be, I would say something like histological subtype, squamous cell or non-squamous would be an example where it's used for stratification, also certain stages of tumors. And then the next level of personalization is what Dr. Singh calls passive personalization. It seems pretty active when you're reviewing these things, but such as the EGFR and targeted therapies. And then the third part is something we're just dealing with, are the so-called active personalized medicine, where you actually generate a different product for each patient. And if you think about it, how are we supposed to regulate a product which is different for every patient? And that's a challenge for us. So I'm going to talk a little bit about, to recoup on the regulatory challenges of biomarker targeted therapies for companion diagnostics when results of an assay are required for safe and effective use of a therapeutic intervention. Then it requires analytical or clinical validation, and these are generally reviewed by CDRH unless they're not. And then we have developed several guidances to either clarify or confuse you depending on how it works. And then the other challenge is the active personalized biomarkers. This, I would say, is evolving on a case-by-case basis because there are preclinical challenges. How do you determine whether there's off-target activity when the target is different in every case? And then autologous manufacture, I think this was discussed earlier, tumor length cell lysates, antigen pulse cells such as the CIP-T Provenge, which was approved a while ago, tumor infiltrating lymphocytes that Dr. Rosenberg's been involved in, then the gene-modified T cells. And I'll just talk about, this was a product of interest a couple of years ago, which was approved, and you will see how the manufacturer requires shipping to an infusion center. And I think we learned a lot of useful lessons from this product, which are applicable to the next generation of cell therapies. And I think Dr. Rosenberg talked about T-cell immunotherapy. I won't cover this other than, as you know, this requires a transduction with the gene transfer vector. And Dr. Porter already covered chimeric antigen receptor T cells, and these again are transduced with either a retrovirus or a lentivirus vector, and some of the challenges thereof. And these are a little bit of the contrast between the engineered T cells and chimeric antigen receptors. And these are some of the targets. We have a target for every tumor, it seems. And unfortunately, some of these are off-target toxicities. And so some of the challenges of these autologous therapies are tracking so you make sure the right patient gets the right product. Believe me, it hasn't always worked out. Transportation, stability, potency, consistency, every product is unique. Manufacturing challenges, manufacturing failures, which have implication for clinical trial design, and also the dose response safety is complex. As Dr. Porter says, it's not clear exactly what the correct dose is, and how do you decide on a dose when these cells are expanding in the patient and they last forever, essentially. And safety concerns, as Dr. Porter mentioned, cytokine release syndrome. These products almost kill the patients, but then they cure them. And so you have to give them a pass. And there's severe neurological toxicity. Some patients have died from these toxicities. Prolonged B-cell aplasia and long-term effects of genetically modified products are unknown. And there's a very high complete response rate, but the long-term effects are unknown. And I'm going to talk briefly about novel combinations. Everybody wants to combine your favorite checkpoint inhibitor with the cancer vaccine du jour. And I think the Cedar Farm Talks person talked about the challenges of whether preclinical tests are needed, and even if preclinical tests are relevant. And vaccine plus checkpoint inhibitor is a typical example of that. And we take it on a case-by-case basis. Then I'll talk a little bit about expedited programs. We've had fast track around for a while, accelerated approval. Dr. Sridhar talked about that, priority review, and breakthrough therapy designation is the new kid on the block. And this was an initiative passed in 2012, and it's drug intended to treat a serious or life-threatening disease or condition, and preliminary clinical evidence indicates that the drug may demonstrate substantial improvement over existing therapies. And I guess that's kind of in the eye of our beholder, but we're developing more experience with that. And as you can see, this is a very busy initiative. And there were 32, last count, 32 breakthrough therapies in CEDAR and 9 in CBER. And I will say that approximately half of those are in the oncology indications. And most recently, we've had 2 oncology products approved, Provenge a couple years ago for the treatment of asymptomatic, minimally symptomatic castrate-resistant prostate cancer. And then just recently, last year, we approved an oncolytic virus, Telimagin Lahaparavec, I think I got that right, or Imlygic. It's an attenuated herpes complex and engineered to express GM-CSF for treatment of melanoma, local treatment of melanoma. So public access to CBER, and these are our emails. And thank you, and I have finished before, just exactly at the red light. I thought that a trap door would open and I would just disappear if I overextended my welcome here. Thank you.